welcome back students. So far we focused only on monosaccharides. Now we are going to study about disaccharides and polysaccharides. We'll study about glycosidic linkage. Actually monosaccharides join together through a bond which is known as glycosidic linkage to form a chain of polysaccharides, or if it has two units of monosaccharide it is disaccharide. So let's start the study of disaccharides and polysaccharides. Let's start. Glycosidic linkage. Linkage between two monosaccharides units through oxygen atom is known as glycosidic linkage. So what happens that monosaccharide units join together through glycosidic linkage to form a chain of monosaccharides. They result in formation of disaccharide, trisaccharide or polysaccharide. So these all are formed by a combination of monosaccharides and they join together through this glycosidic linkage. What happens in glycosidic linkage that let's say these are two monosaccharide units. These come together. They come together and a condensation reaction takes place with elimination of water molecule and a bond is formed here through an oxygen atom, and this is known as glycosidic linkage. Now first is sucrose. Sucrose is our disaccharide which is formed by a combination of D-glucose and D-fructose. So whenever sucrose is hydrolyzed, it results in formation of two units, one glucose unit and one fructose unit, and this is known as invert sugar because the sign of rotation changes from dextro. It was actually dextrorotatory and the products are overall levorotatory. So that is why we can say that inversion is taking place and it is known as invert sugar, and it is a non-reducing sugar and does not have free functional group. You can see from here this is formed by a combination of alpha D glucose and beta D fructose through glycosidic linkage. Next is maltose, which is also known as a malt sugar. It is reducing in nature and it is formed by a combination of glucose units. Next is lactose, which is formed by a combination of beta D galactose and beta D glucose. It is found in milk, so it is called milk sugar also. This is actually reducing half. This beta D glucose is reducing half while beta D galactose is non-reducing half of the sugar. So this was lactose. Now starch is a polysaccharide which is basically a main storage polysaccharide of plant. Plants store their food in form of starch, and it is a polymer of alpha glucose. It consists of long chain of glucose and it is actually of two types. The one which is of straight chain without any branches is known as amylose. You can see from here in amylose alpha D glucose, the D glucose unit, alpha glucose units are linked together in form of a chain. But when there is some side linkage, when two side chains are linked together through some alpha linkage, it is known as amylopectin. So in amylose there is no side linkage with a straight chain only, while in amylopectin two different chains cross-link together. Cellulose is predominant constituent of the cell wall of the plant and it is a straight chain polysaccharide composed of only beta D glucose. Glycogen is basically a storage polysaccharide in animal body. It is found in almost every animal and it is mainly found in kidney and livers where it stores the carbohydrates. It is also known as animal starch because plants store their food in form of starch, animals store their food in form of glycogen, and it is similar in structure to amylopectin. Now there are some chemical tests for carbohydrate. Actually when carbohydrate is heated in dry test tube, it melt, turn brown and finally black. Whenever it is heated in dry tube, first of all the sugar melts, then the melted turns brown and finally black, giving rise to characteristic burning smell of burning sugar, and this is a primary test for carbohydrate. Next is When the carbohydrate is warmed with little concentrated sulfuric acid, it leaves it leaves a residue of carbon which is black, and it is also known as burnt sugar. Next, an important test is Molish test. So far we discussed about physical test. This is Molish. This is a chemical test which involves chemical reaction. In this, there is a Molish reagent which is actually a mixture of alpha naphthol which is dissolved in ethanol. So alpha naphthol dissolved in ethanol is known as Molish reagent. So concentrated sulfuric acid is added to the solution of sugar and Molish reagent. First of all, sugar is mixed with Molish reagent, and to that sulfuric acid is added along the walls of the tube without mixing. A positive reaction is indicated by appearance of purple ring at the interface of the layers formed. So this indicates a positive Molish test. Next is Barfoot test. There is a Barfoot reagent which is a solution of cupric acetate and acetic acid. In this, just say 1 ml sample of solution is placed in a test tube. Solution means the carbohydrate solution is placed in a test tube. In this, 3 ml of Barfoot reagent, which is a sample of cupric acetate and acetic acid, is added. Then the solution is then heated in boiling water bath for 3 minutes, and after those 3 minutes there is a reddish precipitate formed which indicates the presence of carbohydrates. Now there is a solved problem. Starches, yes, it is simply alpha D glucose polymer. Glycogen is similar in its properties, in its structure to amylose pectin. Glycogen is similar in its structure to amylopectin. Now, which of the following is a disaccharide? We know lactose is a disaccharide, but starch is a polysaccharide, cellulose is a polysaccharide, and glucose is a monosaccharide. So the correct option is A here. Hydrolysis of sucrose is called, it is known as inversion, because the optical rotation of reactant and product is totally inversed. So it is known as inversion and not saponification or thermal decomposition or hydration. The correct option is inversion C. Students, in case you have any doubt in this topic, you can post that question in our discussion board. For that, all you need to do is to go to the home page of Ask IITians. There you will find an option of discussion board. You have to click there. Then in the right side, you will find a portion of Ask Experts. In that, just select the category in which you want to post your question. Let's say it's physical chemistry. Then post your question here, and then if you want to attach any image, use this option to attach the image, and finally post it by clicking on Get Answer, and you will be answered within 24 to 48 hours by our experts. So now we are aware of disaccharides and polysaccharides also. In this video, we studied about number of disaccharides like maltose, sucrose, lactose, galactose, and also about polysaccharide like starch and cellulose. With this, we finish with our study of carbohydrates. In next video, we'll start with amino acids. Stay tuned. Thank you.